All right, welcome to the Alaska Sea Life Center, everyone. Thanks for being brave and joining us despite the title of this program today. We're going to be learning about scat, which is fascinating, of course. What we're going to be doing, though, is looking at scat from the angle of science. So how can we learn about scat? And to help us figure that out, we've brought along our resident scat cleaner. Kara here is a research technician. And I'm going to let Kara give a little introduction to what she does and how we collect the scat, what we do with it once we've collected it, and then we'll wrap up in the end with how can we learn from that scat. So, Kara, you want to talk about what you're doing here today?
Yeah, thanks, Darren. So I'm actually a research technician here with the Chiswell Project, which is in fact the longest running research project here at the center. We are looking at the population dynamics of the stellar sea lions in this area. And our main focus is actually during the summer. We have video cameras that we monitor over the summer, watching the pupping season, seeing how that all goes. Throughout the season, though, once winter comes, they start leaving the rookery, the Chiswell rookery where we have our cameras at, and they go to these winter hollowed areas throughout the state and stuff. And that's actually when we do our scat collection. So we have three main haul-out areas that we collect our scat from. Cape Rez, Natoa, and Mary's Bay. We go out, hopefully a couple times each month, January through April, and collect scat at these areas. We take photos of all the sea lions that we see there at the beginning before we jump onto the island and collect the scat. Each scat we put in an individual baggie. So each bag has one scat, hopefully the entire scat of just one sea lion. With that, we can do some hormone analysis, which with the hormones we can usually identify what gender the sea lion is, whether it's male or female. If it's female, we can do pregnancy hormones to see if it's pregnant. And then we can also look at stress hormones. We can also, what I work on right now is actually doing the hard part analysis. So the scat samples that I have already been subsampled for hormones, and I'm now doing the hard part analysis so we can figure out what the sea lions were eating. This is important because winter, is a very hard time, less prey availability, and with the changing climate, everything is changing. So with this, we can actually look throughout the years. We have scat samples all the way from 2013, and we can look and see how their diets have changed over the years to see, maybe how the climate has changed and how that is affecting the sea lions. So I guess we'll just cut to a video that we have taken of me cleaning the scat. So we will take the scat, we soak it in water and Dawn in the baggie, and they're all labeled, so we have to make sure that we keep that same label. We have a coffee filter then that we label as well, so we can make sure that we don't mix samples and everything like that.
Kara, I imagine this process doesn't smell super great.
Oh no. Yes, just imagine walking into the bathroom after someone has eaten at Taco Bell, and then times it by about 100, and that's about how it smells in here. I don't smell it that much, though. Like, after about 10 minutes, you get used to it. Everyone else coming in here, they're like, woo! But I'm used to it. So we will pour the sample out into these sieves. We have 3 different sizes of sifts that we will pour it into. We start with the biggest size, and we want to make sure that we get the entire scat sample out of the baggie. We don't want to miss anything. Once it's completely into the sieve, then we use our fancy, very expensive tools, our sprayer and our paintbrush, to make sure that all the nasty parts are washed away and the hard parts like the bones and everything are left behind. Okay, so once we have 3 different size sieves. So once it gets to the bottom one, which is where this sample I've been working on for a couple hours this morning. This one has a lot of bones and everything, so it's been hard to pick everything out. Once we get to the lowest one, we will then make sure that all these hard parts are kind of into one corner. So then we can dump it into our handy dandy tubber. And we want to try and use the least amount of water, because the more water we pour into our coffee filters, the more likely it is that it's going to break. That's why we have these coffee filter in this also mayonnaise container. So if it does break, then at least the hard parts are still collected in there and we can save those. So after I've dumped most of it into here, you have to make sure that you don't leave any bones behind. If you've ever seen fish bones, they are very small and can get stuck in between these, little places. So I'll just run my finger along the entire back of it to make sure that I don't fill any bones or anything else looking along. And if I feel anything or see anything, I can make sure that I grab it and put it in with the rest of the sample.
Kara, you said you already spent a couple of hours cleaning this one sample today. This seems like a fairly tedious process. What's the average amount of time spent cleaning one particular scat sample, would you say?
If we're lucky, some of them can take as little as 15 minutes. This one has taken almost an hour and a half to do. I've had ones that have lasted up to over 2 hours. So just depending on how many bones there are, how dissolved the scat sample itself is. So a lot of the times, even after soaking in the bags, there's still a lot of clumps that just do not want to, come apart. Once we're pretty confident that we've got everything, one more little bone right there. You can then dump it into our coffee filter.
So while you're doing this, I assume you're trying to minimize the amount of water that you're putting in there.
Yes, while still getting all the bones and everything.
Without losing anything?
Yes.
Ultimately then, after that sample is drained and basically clear of water, where's it going to go next?
So once we have it all drained, we'll actually put it into our drying oven. Which we have one over here. So this is our drying oven, which we keep at 40 degrees Celsius. And we already have some samples here that I've been working on throughout this week. Some of them have been in here for a day, some for a couple days. Usually a sample is dry within a day, but I err on the side of caution and usually keep the samples in our drying oven for at least 2 or 3 days, just because if any little bit of moisture is left in the samples, it will slowly decompose those samples and will make them end up being useless. So we want to make sure that everything is dry so then they can be stored for a long time. And Darren actually has a sample of what we could actually be finding and figure out what's in those.
That's right. So now we're looking through a camera at a cleaned, dried scat sample. And you might notice I am not wearing gloves to work with this sample. This is actually one that's come from our resident stellar sea lions. And Kara has been working with wild stellar sea lion scat. So she has to be very careful about possibly contacting any sort of diseases. But with our resident animals, they have regular health checks. We don't have to be quite as careful. I will definitely wash my hands. Yet again, we're washing our hands plenty these days. But after working with this, but with unknown wild animals, we have to be extremely careful about that. So because this is from a resident animal, we find some more predictable things than we might find out there in the wild. And what I've started doing is just sorting my scat samples. So I'm just basically pulling out pieces that maybe I recognize, or I can even just say, these look similar. And the sorting process is important to be able to start looking through and seeing what kind of things we can identify in there. And once we begin to identify a few parts, we can start to put together a picture of what these sea lions have been eating. Now you notice there's a bunch of large white objects that I'm sort of stacking together here. These are called otoliths. Those are ear stones. And I happen to know those are from a type of cod called walleye pollock, which is also a species of fish that humans eat in large numbers. If you've ever eaten fish sticks, chances are you've had pollock before from here in Alaska. A few other things that I'm finding here. I've got some eye lenses, and these are actually from squid, but there are also fish eye lenses in here, so you can start to tell the difference between different objects like fish eye lenses versus squid eye lenses. I've got some vertebrae. Now, of course, one fish might have several dozen vertebrae, but the otoliths, these ear stones, they really just have pairs of them. And most fish, they'll have 6 total, but there's one pair that is really prominent. And so those are called the sagittae otoliths, and that's what we're looking at here. So really, each pollock would just have a left and a right. Now, if I compare 2 of these in particular, you can see the shapes look exactly the same. These are both from the same side of the body. You can also tell that they're slightly different sizes. And that's an important indication here that these are from 2 different fish because they're, I don't know if they're left or right personally, but scientists would be able to tell. But we know they're both from the same side of the body because they're both the exact same shape. And these things are symmetrical, so one would look like the opposite if it were from the opposite side of the body. But we can start to look at the sizes of these otoliths, and that also indicates the size of the fish because these otoliths grow as the fish grows, and scientists can measure the length of the otolith, and that helps us determine how big the fish was to get an estimate of that. And so we can start to see this animal clearly ate a lot of pollock. There are also a few little otoliths in here from salmon. You can see that the salmon otolith is a very different shape from that pollock otolith. See if I can pick through and find anything else in there. There's a couple little bits of scale inside my scat sample. And in this particular scat sample, I can see some purple glitter. Now, Kara mentioned earlier that we actually do some hormone sampling, and that's one of the things we'll look at with our resident animals. So the hormones are chemicals in the body that regulate and control different processes in the body. So in order to study our resident animals, we will sometimes monitor their hormone levels, and that just tells us how their body is working at any given time. And it can tell us things like if a female is ready to become pregnant or if she is already pregnant. So we can look at those levels. The glitter here then helps us to know whose scat is whose. And I see there's a question in the live chat. What is scat? Scat is just another scientific name for poop. And so we're looking at the remains of what these animals have eaten but it's been processed through their body. So we're looking at digested remains. Oh, and I found, this is like a scavenger hunt, but I found a really fun one there. That's another otolith. And I'm going to zoom in on that one as well as I can because you can see it's vastly different sized from those pollock otoliths. And this one is from a type of fish called a herring, a Pacific herring. You can see it's rather tiny. So just the shape of this otolith tells me the species of fish that the sea lion's been eating. And then again, the size of the otolith can tell us about how big those fish were. So we can really start to learn about their diet. Now, something we might find in there is a squid beak, like this one here. That's another piece that researchers might find. They'll find octopus beaks, squid beaks in scat samples. And all of those pieces can help us put together a picture of the diet of the Steller sea lions. We don't know everything that this animal ate, but we know it ate some pollock. We know it ate at least 1, 2, probably 3, 4 pollock there, some salmon, and some herring. We've got some squid and some fish. And then the vertebrae, experts in other laboratories that we might send these samples to might be able to identify the species of fish from those vertebrae. That's definitely not something that I have the expertise to do, but it is another piece that we're able to identify in there. So the scat analysis portion is something that happens kind of late in the game after Kara has done all the truly burdensome work of cleaning those scat samples and processing them, getting them nice and dry. Those scat samples are preserved for decades, and scientists can go back and look into those scat samples. We can compare the hormone levels that we found before they were subsampled, before Kara got her hands on it, and then know what those animals were eating at various stages of their lives, at various stages of pregnancy, even if we start to look at those hormone levels. If they were stressed, hormones will show that as well, and we can compare their diet to those times of their lives. So it's a really valuable tool. Of course, the animals probably don't mind a whole lot if we're cleaning their rocks for them instead of waiting for the rain to do it. And so Scat is a really valuable scientific tool. It's maybe not the most glamorous of positions to be in, to be the scat cleaner, but I'm sure Kara would tell you it's actually quite fun to come down here into the lab. As long as you get over the smell, it is like a fun little scavenger hunt back here. Something fun is always going on. Now, Kara and I are having to stand 6 feet apart, so she's very awkwardly far away, but I'll let her put her hand in front of the camera from a distance, and we'll say goodbye. Thank you very much for joining us today. Keep track of what's going on our YouTube channel. Find these videos recorded in the Education Corner playlist, and watch our Facebook page as well for other live videos. Thanks again. Take care. Bye-bye.